Cell division and cell cycle mitosis. ChatGPT said. Cell division and cell cycle mitosis. The cell cycle is a series of events that lead to cell division and replication. It consists of interphase and the meiotic phase. The main purpose of the cell cycle is to ensure that one cell divides into two genetically identical daughter cells, maintaining proper function and the integrity of genetic information. Mitosis, a part of the cell cycle, is a process where the nucleus of a cell divides, ensuring the equal distribution of chromosomes to each daughter cell. The cell cycle overview. The cell cycle is divided into two major phases: interphase and meiotic M phase. Interphase. This is the longest phase of the cell cycle, consisting of 3 stages. G1 phase. Gap 1. The cell grows in size, synthesizes proteins, and prepares for DNA replication. It is a phase of intense metabolic activity. The checkpoint here ensures that the cell is ready to proceed to the next phase. S phase. Synthesis. DNA replication occurs in this phase. Each chromosome is duplicated, resulting in 2 sister chromatids attached to the centromere. G2 phase. Gap 2. The cell continues to grow and prepares for mitosis. Additional proteins are synthesized, and the cell checks the integrity of the newly replicated DNA. Meiotic M phase. This phase is where cell division actually occurs. It includes both mitosis, nuclear division, and cytokinesis, cytoplasmic division. Mitosis is a complex process that ensures the division of a single nucleus into 2 genetically identical daughter nuclei. Mitosis. Detailed process. Mitosis is divided into several stages, each with its specific events. The stages of mitosis ensure that the sister chromatids of each chromosome are properly separated and distributed to the daughter cells. Prophase. Chromatin, a complex of DNA and proteins, condenses into visible chromosomes, each consisting of 2 sister chromatids. The nuclear envelope begins to break down. The meiotic spindle, made of microtubules, begins to form. The spindle fibers extend from the centrosomes, which are specialized regions of the cell that organize microtubules. The nucleolus disappears. Chromatophase. The nuclear envelope is completely disassembled. Microtubules from the spindle fibers attach to the centromeres of the chromosomes via a structure called the kinetochore. The chromosomes begin to move toward the center of the cell, aligning along an imaginary plane known as the metaphase plate. Metaphase. Chromosomes align at the metaphase plate in the center of the cell. Each chromosome is attached to microtubules from opposite poles of the cell, ensuring that sister chromatids will be pulled apart in the next phase. Anaphase. The sister chromatids are pulled apart toward opposite poles of the cell as the centromeres are split. This movement is facilitated by the shortening of the microtubules attached to the kinetochores. The cell elongates as microtubules not attached to chromosomes lengthen. Telophase. The separated chromatids, now called chromosomes, reach the opposite poles of the cell. A new nuclear envelope forms around each set of chromosomes. Chromosomes begin to condense back into chromatin, and the nucleolus reappears in each daughter nucleus. Cytokinesis. Although technically not part of mitosis, cytokinesis is the final step in cell division. It involves the division of the cytoplasm and other organelles between the 2 daughter cells. In animal cells, a contractile ring of actin filaments forms, leading to the pinching of the cell membrane cleavage furrow to separate the 2 daughter cells. In plant cells, a cell plate forms between the 2 daughter nuclei, which eventually leads to the formation of a new cell wall. Regulation of the cell cycle. The cell cycle is tightly regulated by checkpoints to ensure that cells only divide when they are ready. Key regulatory proteins include cyclins and cyclin-dependent kinases (CDKs). These proteins activate or deactivate the cell cycle machinery at specific checkpoints. G1 checkpoint restriction point. This checkpoint determines whether the cell should proceed to the S phase. If conditions are unfavorable, the cell enters a resting state called G0. G2 checkpoint ensures that DNA replication is complete and that there are no DNA errors before the cell enters mitosis. M checkpoint spindle checkpoint ensures that chromosomes are properly aligned at the metaphase plate before anaphase begins. If errors are detected, mitosis is halted to prevent aneuploidy, abnormal chromosome number. Importance of mitosis. Mitosis is essential for growth, repair, and asexual reproduction in multicellular organisms. It allows the organism to produce identical cells for tissue maintenance and regeneration. Errors in mitosis can lead to genetic disorders, such as cancer, where cells divide uncontrollably. Key concepts to focus on. Interphase, G1, S, G2 phases. DNA replication occurs in the S phase. Mitosis, prophase, chromatophase, metaphase, anaphase, telophase, chromosome alignment, sister chromatid separation. Cytokinesis, division of the cytoplasm and formation of 2 daughter cells. Regulation, cyclins, CDKs, and checkpoints ensure accurate division. Previous year questions. Question CSIRNET 2019. Which of the following occurs during the anaphase of mitosis? A, chromosomes align at the metaphase plate. B, sister chromatids are pulled apart toward opposite poles. C, the nuclear envelope reforms. D, the nucleolus disappears. Answer, B, sister chromatids are pulled apart toward opposite poles. Question CSIRNET 2020. In which phase of the cell cycle does DNA replication occur? A, G1 phase. B, S phase. C, G2 phase. D, M phase. Answer, B, S phase. Question CSIRNET 2018. Which of the following is true about meiotic division? A, the chromosome number in daughter cells is halved. B, daughter cells are genetically different from the parent cell. C, the process occurs in somatic cells. D, meiosis and mitosis are identical processes. Answer, C, the process occurs in somatic cells. This lecture provides an in-depth exploration of cell division and the cell cycle, focusing on mitosis and its stages. The information is designed to help you grasp key concepts in preparation for the CSIRNET JRF Life Sciences exam, ensuring you're well prepared for related questions. Let me know if you'd like further details or another topic. You said, meiosis, ChatGPT said. Meiosis, detailed process and key concepts. Meiosis is a type of cell division that reduces the chromosome number by half, resulting in 4 non-identical daughter cells, each with half the chromosome number of the original cell. It is essential for sexual reproduction, as it produces gametes, sperm and egg cells in animals and spores in plants. Meiosis consists of 2 consecutive divisions, meiosis I and meiosis II, each with distinct phases, and is crucial for maintaining genetic diversity through processes like crossing over and independent assortment. Overview of meiosis. Meiosis reduces the chromosome number by half and involves 2 rounds of division. Meiosis I, reductional division, where homologous chromosomes are separated. Meiosis II, equational division, where sister chromatids are separated, similar to mitosis. Before meiosis begins, the cell undergoes a preparatory phase called interphase, which includes G1 phase, cell grows and prepares for DNA replication. S phase, DNA replication occurs, resulting in chromosomes consisting of 2 sister chromatids. G2 phase, the cell prepares for meiosis by synthesizing proteins and other molecules. Meiosis I, reductional division prophase I. Chromosomes condense and become visible. Homologous chromosomes pair up in a process known as synapsis. Each pair of homologous chromosomes consists of 4 chromatids, forming a tetrad. Crossing over genetic recombination occurs between non-sister chromatids within a tetrad. This results in the exchange of genetic material, contributing to genetic diversity. The nuclear envelope breaks down, and spindle fibers begin to form. Metaphase I. The tetrads align at the metaphase plate. Homologous chromosomes are attached to spindle fibers from opposite poles, ensuring that each chromosome will be distributed to a different daughter cell. Anaphase I. Homologous chromosomes are separated and pulled to opposite poles. Each chromosome still consists of 2 sister chromatids. Unlike mitosis, sister chromatids are not separated at this stage. Instead, homologous chromosomes, which may carry different genetic variants, are divided, reducing the chromosome number by half. Telophase I. Chromosomes reach the opposite poles of the cell, and the nuclear envelope reforms around each set of chromosomes. Cytokinesis, division of the cytoplasm occurs, resulting in 2 daughter cells, each with half the chromosome number of the original cell haploid. Meiosis II, equational division. Meiosis II is similar to mitosis, but it involves the separation of sister chromatids rather than homologous chromosomes. Prophase II. Chromosomes condense, and a new spindle apparatus forms in each haploid cell. The nuclear envelope disintegrates. Metaphase II. Chromosomes align at the metaphase plate of each haploid cell. The spindle fibers attach to the kinetochores of the sister chromatids. Anaphase II. Before meiosis begins, the cell undergoes a preparatory phase called interphase, which includes G1 phase, cell grows and prepares for DNA replication. S phase, DNA replication occurs, resulting in chromosomes consisting of two sister chromatids. G2 phase, the cell prepares for meiosis by synthesizing proteins and other molecules. Meiosis I, reductional division prophase I. Chromosomes condense and become visible. Homologous chromosomes pair up in a process known as synapsis. Each pair of homologous chromosomes consists of 4 chromatids, forming a tetrad. Crossing over genetic recombination occurs between non-sister chromatids within a tetrad. This results in the exchange of genetic material, contributing to genetic diversity. The nuclear envelope breaks down, and spindle fibers begin to form. Metaphase I. The tetrads align at the metaphase plate. Homologous chromosomes are attached to spindle fibers from opposite poles, ensuring that each chromosome will be distributed to a different daughter cell. Anaphase I. Homologous chromosomes are separated and pulled to opposite poles. Each chromosome still consists of 2 sister chromatids. Unlike mitosis, sister chromatids are not separated at this stage. Instead, homologous chromosomes, which may carry different genetic variants, are divided, reducing the chromosome number by half. Telophase I. Chromosomes reach the opposite poles of the cell, and the nuclear envelope reforms around each set of chromosomes. Cytokinesis division of the cytoplasm occurs, resulting in 2 daughter cells, each with half the chromosome number of the original cell haploid. Meiosis II, equational division. Meiosis II is similar to mitosis, but it involves the separation of sister chromatids rather than homologous chromosomes. Prophase II. Chromosomes condense, and a new spindle apparatus forms in each haploid cell. The nuclear envelope disintegrates. Metaphase II. Chromosomes align at the metaphase plate of each haploid cell. The spindle fibers attach to the kinetochores of the sister chromatids. Anaphase II. The sister chromatids are finally separated and pulled to opposite poles of the cells. Telophase II. Chromatids reach the poles and condense into chromatin. The nuclear envelope reforms around each set of chromosomes, and cytokinesis occurs, resulting in 4 non-identical haploid daughter cells. Key features of meiosis. Reduction in chromosome number. Meiosis reduces the chromosome number by half, ensuring that when 2 gametes fuse during fertilization, the resulting zygote has a correct diploid number of chromosomes. Genetic variation. Meiosis contributes to genetic diversity through crossing over occurs in prophase I, where homologous chromosomes exchange genetic material, leading to recombination of alleles. Independent assortment. During metaphase I, the homologous chromosomes align randomly, ensuring that the combination of chromosomes inherited by the daughter cells is unique. Haploid cells. The final outcome of meiosis is 4 genetically distinct haploid cells, gametes or spores, each with half the number of chromosomes as the original cell. Significance of meiosis. Sexual reproduction. Meiosis is vital for the production of gametes, sperm and egg cells in animals, pollen and ovules in plants. It ensures genetic variation through recombination and independent assortment, which is important for evolution. Prevention of chromosomal abnormalities. By reducing the chromosome number in gametes, meiosis ensures that the fusion of gametes during fertilization restores the diploid chromosome number, preventing chromosome number doubling in every generation. Key concepts to focus on. Meiosis I. Homologous chromosomes are separated, reductional division. Prophase I. Synapsis, crossing over. Anaphase I. Homologous chromosomes are pulled apart. Meiosis II. Sister chromatids are separated, equational division. Similar to mitosis but with haploid cells. Genetic diversity. Crossing over and independent assortment are critical for generating genetic variation. Haploid daughter cells. The end result is 4 genetically diverse haploid cells. Previous year questions. Question CSIRNET 2019. During which stage of meiosis does crossing over occur? A prophase I. Beat metaphase IC, anaphase ID, telophase I. Answer. A prophase I. Question CSIRNET 2020. Which of the following is a consequence of independent assortment during meiosis? A genetic recombination. Beat separation of homologous chromosome C, formation of identical daughter cells D, generation of genetic variation. Answer. D, generation of genetic variation. Question CSIRNET 2018. The haploid number of chromosomes in the cells after meiosis is half of that in the diploid parent cell because of which of the following events? A DNA replication in S phase. Beat separation of homologous chromosomes during meiosis IC, mitosis. Beat separation of sister chromatids during meiosis II. Answer. Beat separation of homologous chromosomes during meiosis I. This lecture provides a comprehensive overview of meiosis, detailing both the stages and the significance of the process in sexual reproduction and genetic diversity. By focusing on the key events, such as crossing over and independent assortment, you'll be well prepared for CSIRNet Life Sciences exam questions related to this topic. Let me know if you'd like another topic or further elaboration. You said. Mitosis and meiosis are regulation. ChatGPT said. Mitosis and meiosis, their regulation. Mitosis and meiosis are tightly regulated processes that ensure accurate cell division, proper chromosome segregation, and the maintenance of genetic stability in organisms. The regulation of these processes is crucial for preventing errors that can lead to diseases like cancer or genetic disorders. Both mitosis and meiosis are controlled by a complex network of checkpoints and regulatory proteins, including cyclins, cyclin-dependent kinases, CDKs, and other associated molecules. Regulation of mitosis. Mitosis is a process of nuclear division in somatic cells, resulting in 2 genetically identical daughter cells. The regulation of mitosis ensures that each cell division occurs correctly, with proper chromosome alignment, segregation, and cytokinesis. The key regulatory mechanisms include the cell cycle checkpoints, cyclins and CDKs, and spindle assembly checkpoints. Cyclins and cyclin-dependent kinases, CDKs. Cyclins are proteins whose levels fluctuate during the cell cycle. Cyclins bind to CDKs, cyclin-dependent kinases, forming complexes that drive the progression through different stages of the cell cycle. Cyclin D and CDK4/6 regulate the transition from the G1 phase to the S phase by promoting DNA replication. Cyclin D and CDK2 regulate the G1S transition. Cyclin A and CDK2 control the S phase and DNA replication. Cyclin B and CDK1, also known as CDC2, regulate the entry into mitosis M phase. This complex is essential for the transition from G2 to M phase and helps coordinate the steps of mitosis. Regulation of mitosis through checkpoints. G1 checkpoint restriction point before entering the S phase. The cell checks for DNA damage and whether the environment is favorable for DNA replication. If there is DNA damage, the cell will arrest at this point, allowing repair or apoptosis to occur. G2 checkpoint. Before mitosis begins, the cell checks for DNA replication errors and ensures that the DNA is undamaged. If any issues are found, the cell cycle is halted to allow repair mechanisms to fix the problems. M checkpoint, spindle checkpoint. This checkpoint ensures that chromosomes are properly attached to the meiotic spindle before anaphase. If the chromosomes are not aligned properly at the metaphase plate or if spindle fibers are not attached correctly to kinetochores, the cell will not proceed to anaphase, preventing chromosome segregation. Anaphase promoting complex APCC. The APCC is a ubiquitin ligase that targets regulatory proteins for degradation, ensuring the progression of mitosis. It marks securin and cyclin B for degradation, allowing sister chromatids to separate during anaphase. Spindle assembly checkpoint, SAC. The SAC ensures that the meiotic spindle is correctly assembled and the chromosomes are correctly aligned. It prevents the activation of APCC until all chromosomes are properly attached to the spindle fibers. Regulation of meiosis. Meiosis consists of 2 rounds of cell division, meiosis I and meiosis II, and is essential for producing haploid gametes, sperm and eggs in animals and spores in plants. The regulation of meiosis is essential to maintain chromosome integrity and genetic diversity. The regulation involves similar checkpoints as mitosis, but there are also meiosis-specific mechanisms that govern recombination and segregation of chromosomes. Regulation by cyclins and CDKs. Like mitosis, cyclins and CDKs regulate key transitions in meiosis. The key difference in meiosis is that cyclins are involved in both the initiation of meiosis and the arresting of the cell cycle at specific stages. Cyclin B and CDK1 regulate the initiation of meiosis, particularly during prophase I, when homologous chromosomes undergo recombination. Cyclin A and CDK2 regulate progression through meiosis, especially during the transition from metaphase I to anaphase I. Regulation of meiosis I. Prophase I is the most complex phase in meiosis, as it involves homologous recombination, crossing over synapsis, pairing of homologous chromosomes, and the formation of a synaptonemal complex. The regulation of these processes ensures genetic diversity. SPO11. This protein introduces double-strand breaks in the DNA, which are necessary for homologous recombination. Checkpoint during prophase I. The checkpoint ensures that homologous chromosomes are properly paired and recombined before progressing to metaphase I. Meiosis I checkpoints. Synapsis checkpoint. This checkpoint ensures that homologous chromosomes are properly paired and recombined. If there is an error in synapsis or recombination, the cell may undergo apoptosis or arrest at the checkpoint. Spindle assembly checkpoint. Like mitosis, meiosis I has a spindle assembly checkpoint that ensures that the chromosomes are correctly aligned at the metaphase plate before anaphase I. This prevents errors such as aneuploidy. Meiosis II regulation. Meiosis II is similar to mitosis but involves the separation of sister chromatids rather than homologous chromosomes. Cyclin D and CDK1 play a crucial role in regulating the transition from metaphase II to anaphase II by ensuring that sister chromatids are correctly aligned before separation. The stacked spindle assembly checkpoint also plays a critical role in ensuring that the sister chromatids are attached to the spindle fibers and aligned before proceeding to anaphase II. Key differences in the regulation of mitosis and meiosis chromosome number. Mitosis results in 2 genetically identical diploid daughter cells, while meiosis results in 4 genetically diverse haploid cells. The regulatory mechanisms in meiosis ensure that homologous chromosomes are segregated in meiosis I and that sister chromatids are separated in meiosis II, maintaining the correct chromosome number in the gametes. Crossing over. Crossing over in meiosis adds genetic diversity by exchanging genetic material between homologous chromosomes. This occurs during prophase I of meiosis I and is tightly regulated by proteins such as SPO11. Spindle assembly checkpoint. Both mitosis and meiosis involve a spindle assembly checkpoint to ensure proper chromosome alignment. However, the sac is more complex in meiosis, especially due to the need for homologous chromosome pairing and the prevention of non-disjunction during meiosis I. Cell cycle arrest. In meiosis, there are 2 key points where the cell cycle can arrest: during prophase I, where the cell can be arrested until recombination is completed, and during metaphase II, where the cell halts until the chromosomes are properly aligned. Previous year questions. Question CSIRNET 2019. Which of the following proteins are involved in regulating the G1S checkpoint in the cell cycle? Cyclin B and CDK1. Beat cyclin D and CDK46C, cyclin A and CDK2D, cyclin D and CDK2. Answer: Beat cyclin D and CDK46. Question CSIRNET 2020. In meiosis, what is the consequence of a spindle assembly checkpoint sac failure during anaphase I? Homologous chromosomes segregate randomly. Beat sister chromatids segregate to opposite poles. C. There is an unequal segregation of chromosomes, leading to aneuploidy. D. Crossing over is inhibited. Answer: C. There is an unequal segregation of chromosomes, leading to aneuploidy. Question CSIRNET 2021. What role does CDK1 play in regulating mitosis? It prevents chromosome condensation. B. It regulates DNA replication during S phase. C. It activates cyclin B to trigger the onset of mitosis. D. It promotes cytokinesis. Answer: C. It activates cyclin B to trigger the onset of mitosis. This lecture has outlined the regulation of mitosis and meiosis, covering the key checkpoints, cyclins, and CDKs that control the progression of the cell cycle and ensure proper chromosome segregation. This information is crucial for understanding the molecular mechanisms behind cell division and for preparing for CSIRNet life sciences exams. You said. Steps in cell cycle. ChatGPT said. Steps in the cell cycle. The cell cycle is a series of events that lead to cell division and the production of 2 daughter cells. It consists of 2 major phases: interphase and M phase mitosis. The cell cycle is tightly regulated to ensure proper cell division, DNA replication, and the maintenance of genomic integrity. The primary steps in the cell cycle include 1. Interphase. Interphase is the longest phase of the cell cycle, during which the cell grows, carries out its normal functions, and prepares for cell division. Interphase is subdivided into 3 distinct phases: G1 phase, gap 1, S phase, synthesis, and G2 phase, gap 2. G1 phase, gap 1. This phase is characterized by the cell's growth and the synthesis of proteins necessary for DNA replication. The cell increases in size, produces RNA, and synthesizes proteins required for DNA replication. The G1 checkpoint, also known as the restriction point, checks for DNA damage and ensures that the environment is favorable for DNA replication. If conditions are unfavorable, the cell may enter a resting phase called G0, where it exits the cell cycle. If conditions are favorable, the cell moves into the S phase. G1 phase, gap 1. This phase is characterized by the cell's growth and the synthesis of proteins necessary for DNA replication. The cell increases in size, produces RNA, and synthesizes proteins required for DNA replication. The G1 checkpoint, also known as the restriction point, checks for DNA damage and ensures that the environment is favorable for DNA replication. If conditions are unfavorable, the cell may enter a resting phase called G0, where it exits the cell cycle. If conditions are favorable, the cell moves into the S phase. S phase, synthesis. The S phase is where DNA replication occurs. Each chromosome is duplicated, resulting in 2 sister chromatids held together at the centromere. The replication ensures that each daughter cell will receive an identical set of chromosomes after division. DNA synthesis is regulated by various cyclins and CDKs, which control the progression of the cell through the S phase. G2 phase, gap 2. During the G2 phase, the cell continues to grow and prepare for mitosis. The cell synthesizes proteins required for cell division and checks for any DNA replication errors that may have occurred in the S phase. The G2 checkpoint ensures that the cell has fully replicated its DNA and that there are no errors before proceeding to mitosis. The transition from G1 to S phase and from G2 to M phase is regulated by cyclin CDK complexes, which ensure that the cell cycle progresses only when the appropriate conditions are met. 2. M phase mitosis. Mitosis is the phase during which the cell divides into 2 genetically identical daughter cells. Mitosis is divided into several stages, each carefully regulated to ensure accurate chromosome segregation. Prophase. During prophase, the chromatin condenses into visible chromosomes, each consisting of 2 sister chromatids connected by a centromere. The nuclear envelope begins to break down, and the meiotic spindle, which is composed of microtubules, begins to form. The centrosomes organelles responsible for organizing microtubules start moving to opposite poles of the cell, and the spindle fibers begin to extend towards the chromosomes. Prometaphase. The nuclear envelope fully disassembles, and the spindle fibers attach to the kinetochores protein structures at the centromeres of chromosomes. Chromosomes begin to move towards the metaphase plate, the equatorial plane of the cell. Metaphase. In metaphase, the chromosomes align along the metaphase plate, ensuring that each sister chromatid is attached to spindle fibers from opposite poles. The metaphase checkpoint ensures that all chromosomes are correctly attached to the spindle before proceeding to anaphase. Anaphase. During anaphase, the sister chromatids are pulled apart towards opposite poles of the cell as the centromere splits. This separation is facilitated by the shortening of the spindle fibers attached to the chromatids, ensuring that each daughter cell receives an identical set of chromosomes. Telophase. Telophase is the final stage of mitosis, during which the separated chromatids condense back into chromatin. The nuclear envelope reforms around each set of chromosomes, creating 2 distinct nuclei within the cell. The spindle apparatus disassembles. Cytokinesis. Cytokinesis is the process of cytoplasmic division that occurs after mitosis. In animal cells, a contractile ring made of actin filaments forms, leading to the pinching of the cell membrane and the formation of 2 daughter cells. In plant cells, a new cell wall, the cell plate, forms between the 2 daughter cells. The successful completion of mitosis results in the formation of 2 genetically identical daughter cells. 3. Regulation of the cell cycle. The cell cycle is tightly regulated by a series of checkpoints that ensure proper progression through each phase. Key regulatory molecules include cyclins. These are proteins whose levels fluctuate during the cell cycle. They bind to cyclin-dependent kinases, CDKs, to regulate the progression through the cell cycle. CDKs, cyclin-dependent kinases, are enzymes that, when activated by binding to cyclins, phosphorylate other proteins to drive the cell cycle forward. Cyclin CDK complexes. These complexes control transitions between cell cycle phases by phosphorylating target proteins that promote progression, e.g., cyclin B-CDK1 complex for mitosis. Checkpoints. The cell cycle includes checkpoints at G1, G2, and M phases, where the cell checks for DNA damage, replication errors, or incomplete division before progressing. Tumor suppressors e.g., p53. Tumor suppressors such as p53 can halt the cell cycle in response to DNA damage, allowing for repair or triggering apoptosis if the damage is irreparable. Proto-oncogenes e.g., cyclin D. Proto-oncogenes promote cell cycle progression. Mutations in proto-oncogenes can lead to uncontrolled cell division and cancer. Previous year questions. Question CSIRNET 2018. Which of the following is the correct sequence of events in the cell cycle? A. G1 right pointing arrow S right pointing arrow G2 right pointing arrow M. B. S right pointing arrow G1 right pointing arrow G2 right pointing arrow M. C. G1 right pointing arrow G2 right pointing arrow S right pointing arrow M. D. M right pointing arrow S right pointing arrow G1 right pointing arrow G2. Answer. A. G1 right pointing arrow S right pointing arrow G2 right pointing arrow M. Question CSIRNET 2020. During which phase of the cell cycle is the DNA content doubled? A. G1 phase. B. S phase. C. G2 phase. D. M phase. Answer. B. S phase. Question CSIRNET 2021. Which protein complex is responsible for the initiation of mitosis? A. Cyclin D. CDK4. B. Cyclin E. CDK2. C. Cyclin B. CDK1. D. Cyclin A. CDK2. Answer. C. Cyclin B. CDK1. This lecture provides a comprehensive overview of the steps in the cell cycle, from This lecture provides a comprehensive overview of the steps in the cell cycle, from interphase through mitosis, with a focus on the key regulatory proteins and checkpoints involved. Understanding these processes is crucial for preparation for the CSIRNet Life Sciences exam. You said. Regulation and control of cell cycle. ChatGPT said. Regulation and control of the cell cycle. The cell cycle is a tightly regulated process that ensures the proper division and proliferation of cells. This regulation is crucial for maintaining cellular integrity and preventing diseases such as cancer. The cell cycle is controlled by a complex network of signaling pathways involving various proteins and checkpoints that ensure the fidelity of DNA replication, chromosome segregation, and cell division. Key regulators of the cell cycle. Cyclins. Cyclins are regulatory proteins that are synthesized and degraded at specific stages of the cell cycle. They bind to and activate cyclin-dependent kinases, CDKs, which are enzymes that drive the progression of the cell cycle. Cyclins exist in different types, each associated with specific phases of the cell cycle. Cyclin D acts during the G1 phase and is involved in the G1 to S phase transition. Cyclin E present during late G1 and early S phase, regulating the G1 S checkpoint. Cyclin A functions in the S and G2 phases, ensuring DNA replication and preparation for mitosis. Cyclin B present during G2 and M phases, necessary for the transition from G2 to M phase entry. Cyclin-dependent kinases, CDKs are enzymes that, when bound to cyclins, phosphorylate specific target proteins to trigger the progression of the cell cycle. The activity of CDKs is regulated by the availability of cyclins and their binding to cyclin CDK complexes. CDKs must be activated by cyclins to phosphorylate proteins involved in key events such as DNA replication, chromosome condensation, and spindle formation. CDK inhibitors, CKIs, cyclin-dependent kinase inhibitors, CKIs, are proteins that regulate the activity of cyclin CDK complexes. They act as negative regulators of the cell cycle, preventing cell cycle progression when needed. The two major families of CKIs are INK4 family, e.g., P16Q4 inhibits CDK4 and CDK6, preventing the G1 to S phase transition. CYP family, e.g., P21, P27. These CKIs inhibit a broad range of cyclin CDK complexes and are involved in multiple checkpoints, including G1S and G2M. Retinoblastoma protein, RB. The retinoblastoma protein, RB, is a key tumor suppressor that regulates the G1S checkpoint. RB controls the activity of the transcription factor E2F, which is essential for the expression of genes required for DNA replication. In its hyperphosphorylated form, RB binds to E2F and prevents transcription. When RB is phosphorylated by cyclin D CDK4/6 and cyclin E CDK2 complexes, it releases E2F, allowing the cell to progress from G1 to S phase. P53 tumor suppressor. P53 is a critical tumor suppressor protein that acts as a guardian of the genome. It is activated in response to DNA damage and can induce cell cycle arrest at the G1S checkpoint by increasing the levels of P21, a CDK inhibitor. If the damage is irreparable, P53 can initiate apoptosis to prevent the proliferation of damaged cells. P53 plays a major role in preventing the accumulation of mutations that could lead to cancer. P21. P21 is a cyclin-dependent kinase inhibitor, CKI, regulated by P53. Upon activation of P53 in response to DNA damage, P21 inhibits cyclin CDK complexes, particularly cyclin D CDK4/6 and cyclin E CDK2, thus halting the cell cycle to allow DNA repair. P21 also contributes to the G1S checkpoint by blocking the activation of cyclin CDK complexes. Major cell cycle checkpoints. The cell cycle is regulated by several checkpoints that monitor the integrity of the cell and its environment. These checkpoints are essential for ensuring that cells do not proceed to the next stage until the previous phase is completed correctly. If DNA damage or replication errors are detected, the checkpoints can delay progression or induce apoptosis. G1S checkpoint restriction point. The G1S checkpoint is one of the most critical points in the cell cycle, as it determines whether the cell will proceed to DNA replication. This checkpoint is regulated by cyclins, cyclin D, cyclin E, and CDKs, which act in concert with tumor suppressors like RB and P53. If the cell has sufficient nutrients, growth factors, and no DNA damage, the cell progresses into the S phase. However, if there is DNA damage, the checkpoint activates P53, which induces P21 to inhibit CDK activity, halting progression into S phase. G2M checkpoint. The G2M checkpoint ensures that DNA has been replicated correctly and that the cell is ready to enter mitosis. Cyclin B CDK1 complexes are critical at this checkpoint. If DNA damage or incomplete DNA replication is detected, the checkpoint will delay mitosis to allow for repair. In the case of irreparable damage, the checkpoint may initiate apoptosis. M checkpoint. Spindle assembly checkpoint. The M checkpoint occurs during mitosis, before anaphase, and ensures that all chromosomes are correctly attached to the spindle fibers. If any chromosome is not properly aligned or attached, the checkpoint prevents the progression to anaphase, ensuring that chromosome segregation is accurate. The anaphase promoting complex APC is activated once all chromosomes are properly aligned, leading to the degradation of securin and the activation of separase, allowing sister chromatids to be separated. External and internal signals affecting cell cycle progression. Growth factors. External signals, such as growth factors e.g., EGF, PDGF bind to receptors on the cell surface and activate intracellular signaling cascades e.g., MEPK pathway. These signals stimulate the expression of cyclins and other cell cycle regulators, promoting cell cycle progression. DNA damage response. DNA damage induces the activation of the ATM/ATR kinases, which phosphorylate and activate P53. Activated P53 then triggers cell cycle arrest by inducing P21 expression. If the damage is too severe to repair, P53 can also activate apoptotic pathways. Telomere shortening. As cells divide, telomeres shorten, which can act as a signal to arrest the cell cycle. Shortened telomeres are recognized by the DNA damage response machinery, leading to P53 activation and cell cycle arrest in G1 or G2. Nutrient availability. Cells monitor the availability of nutrients, such as amino acids and oxygen, before committing to cell division. Insufficient nutrients can trigger cell cycle arrest at the G1 phase. Oncogenes and tumor suppressors. Oncogenes are mutated or overexpressed versions of normal genes, proto-oncogenes, that drive the cell cycle inappropriately, leading to uncontrolled cell division. Examples include cyclin D, ROS, and MYC. Tumor suppressor genes, such as P53, RB, and P10, normally inhibit cell cycle progression and prevent tumor formation. Loss of function in these tumor suppressors leads to unregulated cell division and tumorigenesis. Previous year questions. Question CSIRNET 2018. Which of the following is the function of P53 in the regulation of the cell cycle? Inhibits cyclin CDK complexes during the G1 phase. B activates cyclin CDK complexes during the S phase. C promotes DNA replication during the S phase. D regulates the separation of chromatids during anaphase. Answer. Inhibits cyclin CDK complexes during the G1 phase. Question CSIRNET 2020. Which of the following is a key regulator of the G1S checkpoint? A. P53. B. Cyclin BCDK1. C. Cyclin ECDK2. D. Cyclin ACDK1. Answer. C. Cyclin ECDK2. Answer. C. Cyclin ECDK2. Question CSIRNET 2021. Previous year questions. Question CSIRNET 2018. Which of the following is a function of P53 in the regulation of the cell cycle? A. Inhibits cyclin CDK complexes during the G1 phase. B. Activates cyclin CDK complexes during the S phase. C. Promotes DNA replication during the S phase. D. Regulates the separation of chromatids during anaphase. Answer. A. Inhibits cyclin CDK complexes during the G1 phase. Question CSIRNET 2020. Which of the following is a key regulator of the G1S checkpoint? A. P53. B. Cyclin BCDK1. C. Cyclin ECDK2. D. Cyclin ACDK1. Answer. C. Cyclin ECDK2. Question CSIRNET 2021. What is the primary function of the anaphase promoting complex APC in cell cycle regulation? A. Induces DNA repair mechanisms. B. Triggers the separation of sister chromatids. C. Activates cyclin CDK complexes. D. Prevents DNA replication in S phase. Answer. B. Triggers the separation of sister chromatids. This lecture covers the regulation and control of the cell cycle, including the key regulatory molecules, checkpoints, and signaling pathways involved in ensuring the proper progression of the cell cycle. Understanding these mechanisms is essential for preparation for the CSIRNet Life Sciences exam. You said. Microbial physiology. ChatGPT said. Microbial physiology. Microbial physiology is the study of the functions and processes that occur within microorganisms, including bacteria, fungi, algae, and protozoa. It focuses on the biochemical and molecular mechanisms that allow these organisms to grow, reproduce, and respond to their environment. Microbial physiology encompasses various aspects such as metabolism, energy production, nutrient utilization, growth, and stress response. Understanding microbial physiology is essential for applications in medicine, agriculture, environmental microbiology, and biotechnology. 1. Cellular structure and function in microorganisms. Microorganisms, although diverse in their shape and structure, share common cellular components. Cell wall. Many microorganisms, especially bacteria, have a cell wall that provides structural support and protects the cell from osmotic pressure. In bacteria, the composition of the cell wall peptidoglycan determines the gram-staining property, gram-positive or gram-negative. Plasma membrane. The plasma membrane is a selective barrier that regulates the entry and exit of molecules. It also contains proteins involved in transport, signaling, and energy production. Cytoplasm. The cytoplasm contains the cellular machinery for metabolism, protein synthesis, and other essential functions. In bacteria, the cytoplasm also houses the nucleoid, where the DNA is located. Nucleoid. In prokaryotes, the nucleoid is a region within the cytoplasm where the genetic material, DNA, resides. In contrast, eukaryotic microorganisms have membrane-bound nuclei. Ribosomes. Ribosomes in microorganisms are responsible for protein synthesis. They are composed of ribosomal RNA, rRNA, and proteins that are found either free in the cytoplasm or attached to the plasma membrane. 2. Microbial metabolism. Microbial metabolism involves all the biochemical reactions that occur in microorganisms, from energy production to the synthesis of cellular components. Metabolic pathways in microorganisms are classified as either catabolic, breaking down molecules to release energy, or anabolic, using energy to synthesize molecules. Catabolism. The breakdown of complex molecules into simpler ones, releasing energy in the form of ATP, adenosine triphosphate. This energy is used for cellular processes. Fermentation. Anaerobic breakdown of glucose or other organic compounds. For example, in yeast, glucose undergoes fermentation to produce ethanol and carbon dioxide. Respiration. The complete oxidation of organic compounds, e.g., glucose to produce carbon dioxide, water, and ATP. This can be aerobic, requiring oxygen, or anaerobic, occurring without oxygen. Glycolysis. The breakdown of glucose to pyruvate, generating ATP and NADH. This process occurs in the cytoplasm. Citric acid cycle, Krebs cycle, occurs in the mitochondria, in eukaryotes, or the cytoplasm, in prokaryotes. It completes the oxidation of glucose derivatives and generates ATP, NADH, and FADH2. Two. Microbial metabolism. Microbial metabolism involves all the biochemical reactions that occur in microorganisms, from energy production to the synthesis of cellular components. Metabolic pathways in microorganisms are classified as either catabolic, breaking down molecules to release energy, or anabolic, using energy to synthesize molecules. Catabolism. The breakdown of complex molecules into simpler ones, releasing energy in the form of ATP, adenosine triphosphate. This energy is used for cellular processes. Fermentation. Anaerobic breakdown of glucose or other organic compounds. For example, in yeast, glucose undergoes fermentation to produce ethanol and carbon dioxide. Respiration. The complete oxidation of organic compounds, e.g., glucose to produce carbon dioxide, water, and ATP. This can be aerobic, requiring oxygen, or anaerobic, occurring without oxygen. Glycolysis. The breakdown of glucose to pyruvate, generating ATP and NADH. This process occurs in the cytoplasm. Citric acid cycle, Krebs cycle, occurs in the mitochondria, in eukaryotes, or the cytoplasm, in prokaryotes. It completes the oxidation of glucose derivatives and generates ATP, NADH, and FADH2. Electron transport chain, etc. The final stage of respiration, where electrons are transferred through protein complexes embedded in the membrane, generating a proton gradient that drives ATP synthesis. Anabolism. The synthesis of complex molecules from simpler ones, using energy. Key anabolic pathways include protein synthesis, the assembly of amino acids into proteins, which is a vital anabolic process in microbial cells. Nucleotide synthesis, the creation of nucleotides for DNA and RNA synthesis. Cell wall biosynthesis, the synthesis of peptidoglycan in bacteria or chitin in fungi for the formation of the cell wall. 3. Energy production in microorganisms. Energy production is a critical aspect of microbial physiology. Microorganisms have evolved various pathways to generate ATP, which is used as an energy currency for cellular processes. Aerobic respiration. In the presence of oxygen, microorganisms oxidize glucose through glycolysis, the citric acid cycle, and the electron transport chain, yielding a large amount of ATP. Anaerobic respiration. When oxygen is unavailable, many microorganisms switch to anaerobic respiration, using alternate electron acceptors such as nitrate, sulfate, or carbon dioxide. Fermentation. In the absence of oxygen or when oxygen is insufficient, fermentation pathways are used to regenerate NAD+ and produce small amounts of ATP. Lactic acid fermentation, e.g., in muscle cells, and alcoholic fermentation, e.g., in yeast, are two common types. 4. Microbial growth and growth phases. Microbial growth refers to an increase in the number of cells. The growth of microorganisms is influenced by factors such as nutrient availability, temperature, pH, and oxygen levels. Growth phases. Microbial growth is typically characterized by a series of phases. Lag phase. The initial phase where cells are adapting to the new environment and preparing for growth. Exponential phase, also called the log phase, during which cells divide at a constant rate, leading to an exponential increase in population size. Stationary phase. In this phase, the rate of cell division equals the rate of cell death due to depletion of nutrients or accumulation of waste products. Death phase. The population decreases as cells die off at a faster rate than they divide. Growth rate and generation time. The rate at which microorganisms grow can be measured by their doubling time or generation time, the time it takes for the population to double. 5. Microbial stress response. Microorganisms often face stressful conditions such as nutrient limitation, temperature extremes, or exposure to toxic compounds. They have evolved various mechanisms to survive these conditions. Heat shock proteins. Microorganisms produce heat shock proteins, HSPs, in response to elevated temperatures. These proteins help in refolding denatured proteins and maintaining cellular integrity. Osmotic stress. Microorganisms exposed to hypertonic or hypotonic environments produce osmotic regulators like triolos or glycerol to protect cellular components. Acid and alkaline stress. Microorganisms can adjust their internal pH by pumping protons in or out of the cell or synthesizing buffering agents. 6. Microbial signaling and communication. Many microorganisms can communicate with each other through processes such as quorum sensing, which allows them to coordinate group behaviors, such as biofilm formation or the production of virulence factors. Quorum sensing involves the release and detection of signaling molecules called autoinducers. 7. Microbial adaptations and genetic regulation. Microorganisms exhibit genetic flexibility that allows them to adapt to changing environments. This includes gene regulation. Bacteria can regulate the expression of specific genes in response to environmental signals, often through operons like the LAC operon or the TRP operon. Horizontal gene transfer. Microorganisms can exchange genetic material through processes such as transformation, transduction, and conjugation, facilitating rapid adaptation to new environments or the acquisition of antibiotic resistance. Previous year questions. Question CSIRNET 2018. In bacteria, the primary role of the electron transport chain is to generate ATP. B. Convert glucose into pyruvate C. Catalyze protein synthesis D. Synthesize lipids. Answer. Generate ATP. Question CSIRNET 2020. Which of the following is a product of alcoholic fermentation in yeast? A. Lactic acid. B. Ethanol and carbon dioxide. C. Acetate. D. Oxygen. Answer. B. Ethanol and carbon dioxide. Question CSIRNET 2021. Which of the following is involved in the regulation of the LAC operon in E. coli? A. Repressor protein and lactose. B. Cyclic AMP and glucose C. Promoter and ribosome D. All of the above. Answer. A. Repressor protein and lactose. This lecture provides a comprehensive overview of microbial physiology, including metabolism, growth, stress response, and genetic regulation. Previous year questions. Question CSIRNET 2018. In bacteria, the primary role of the electron transport chain is to a. Generate ATP. B. Convert glucose into pyruvate C. Catalyze protein synthesis D. Synthesize lipids. Answer. A. Generate ATP. Question CSIRNET 2020. Which of the following is a product of alcoholic fermentation in yeast? A. Lactic acid. B. Ethanol and carbon dioxide. C. Acetate. D. Oxygen. Answer. B. Ethanol and carbon dioxide. Question CSIRNET 2021. Which of the following is involved in the regulation of the LAC operon in E. coli? A. Repressor protein and lactose. B. Cyclic AMP and glucose C. Promoter and ribosome D. All of the above. Answer. A. Repressor protein and lactose. This lecture provides a comprehensive overview of microbial physiology, including metabolism, growth, stress response, and genetic regulation. It is crucial for understanding the diverse ways microorganisms survive, grow, and adapt to their environments. These concepts are key for exam preparation, especially for the CSIRNET Life Sciences exam. You said. Growth yield and characteristics. ChatGPT said. Growth yield and characteristics in microbial physiology. In microbial physiology, growth yield refers to the amount of cellular biomass produced by microorganisms in relation to the substrate or nutrients consumed. It is a key indicator of microbial efficiency and is essential for understanding microbial metabolism, growth patterns, and the factors affecting microbial productivity. Growth characteristics, on the other hand, describe the patterns and behavior of microorganisms during their growth phases, helping to explain how microorganisms utilize nutrients and energy under various environmental conditions. 1. Growth yield. The growth yield is often quantified as the ratio of microbial biomass measured in terms of dry weight or cell number to the amount of substrate, typically carbon sources like glucose, consumed during growth. This ratio provides insights into the efficiency of microbial growth under specific conditions. Yields in different conditions. Growth yield can vary depending on whether microorganisms are grown under aerobic or anaerobic conditions, with aerobic growth typically yielding higher biomass production due to the complete oxidation of nutrients. Substrate utilization. The yield is also dependent on the type of carbon source used. For instance, glucose tends to have a higher yield compared to other complex sugars or organic compounds, as it is readily metabolized. Energy efficiency. The higher the growth yield, the more efficient the microorganism is at converting available nutrients into biomass, which is often influenced by the energy available in the metabolic pathways used. Formula for growth yield YXS. The growth yield is mathematically expressed as Y. XS equals amount of biomass produced X amount of substrate consumed SY. X. S equals amount of substrate consumed S amount of biomass produced X. Example, if 1 mole of glucose produces 0.5 moles of biomass X, the yield is 0.5 moles of biomass per mole of glucose consumed. 2. Growth characteristics. Microbial growth is often studied in terms of the growth curve, which outlines the phases of microbial population growth and how microorganisms adapt to environmental changes. The classic microbial growth curve consists of the following stages. Lag phase. In this initial phase, microorganisms are metabolically active but not dividing. They are adjusting to the environment, synthesizing the necessary enzymes, and preparing for active growth. The duration of this phase depends on the initial inoculum, media composition, and the physiological state of the cells. Exponential log phase. During the exponential phase, microorganisms divide at a constant rate, leading to an exponential increase in the number of cells. This phase represents the period of maximal growth, where cells are actively synthesizing macromolecules like proteins, lipids, and nucleic acids. The rate of growth is highly dependent on the availability of nutrients, temperature, and pH. The specific growth rate, measured in generation time, the time it takes for the population to double, is highest during this phase. Stationary phase. As nutrients become depleted and waste products accumulate, the growth rate slows down and eventually stabilizes. The rate of cell division equals the rate of cell death. During this phase, microorganisms undergo metabolic adjustments to cope with nutrient scarcity. Some microorganisms may switch to secondary metabolic pathways, producing antibiotics or other protective compounds. Stationary phase is often marked by a balance between growth and death. Death decline phase. In the death phase, the number of dying cells exceeds the number of new cells being formed. This phase is characterized by nutrient depletion, accumulation of toxic waste products, and a general decline in cellular functions. Microorganisms that cannot adapt to the stressful conditions eventually die. 2. Growth characteristics. Microbial growth is growth yield and characteristics in microbial physiology. In microbial physiology, growth yield refers to the amount of cellular biomass produced by microorganisms in relation to the substrate or nutrients consumed. It is a key indicator of microbial efficiency and is essential for understanding microbial metabolism, growth patterns, and the factors affecting microbial productivity. Growth characteristics, on the other hand, describe the patterns and behavior of microorganisms during their growth phases, helping to explain how microorganisms utilize nutrients and energy under various environmental conditions. 1. Growth yield. The growth yield is often quantified as the ratio of microbial biomass measured in terms of dry weight or cell number to the amount of substrate, typically carbon sources like glucose, consumed during growth. This ratio provides insights into the efficiency of microbial growth under specific conditions. Yields in different conditions. Growth yield can vary depending on whether microorganisms are grown under aerobic or anaerobic conditions, with aerobic growth typically yielding higher biomass production due to the complete oxidation of nutrients. Substrate utilization. The yield is also dependent on the type of carbon source used. For instance, glucose tends to have a higher yield compared to other complex sugars or organic compounds, as it is readily metabolized. Energy efficiency. The higher the growth yield, the more efficient the microorganism is at converting available nutrients into biomass, which is often influenced by the energy available in the metabolic pathways used. Formula for growth yield YXS. The growth yield is mathematically expressed as Y. XS equals amount of biomass produced X amount of substrate consumed SY. X. S equals amount of substrate consumed S amount of biomass produced X. Example, if 1 mole of glucose produces 0.5 moles of biomass X, the yield is 0.5 moles of biomass per mole of glucose consumed. 2. Growth characteristics. Microbial growth is often studied in terms of the growth curve, which outlines the phases of microbial population growth and how microorganisms adapt to environmental changes. The classic microbial growth curve consists of the following stages. Lag phase. In this initial phase, microorganisms are metabolically active but not dividing. They are adjusting to the environment, synthesizing the necessary enzymes, and preparing for active growth. The duration of this phase depends on the initial inoculum, media composition, and the physiological state of the cells. Exponential log phase. During the exponential phase, microorganisms divide at a constant rate, leading to an exponential increase in the number of cells. This phase represents the period of maximal growth, where cells are actively synthesizing macromolecules like proteins, lipids, and nucleic acids. The rate of growth is highly dependent on the availability of nutrients, temperature, and pH. The specific growth rate, measured in generation time, the time it takes for the population to double, is highest during this phase. Stationary phase. As nutrients become depleted and waste products accumulate, the growth rate slows down and eventually stabilizes. The rate of cell division equals the rate of cell death. During this phase, microorganisms undergo metabolic adjustments to cope with nutrient scarcity. Some microorganisms may switch to secondary metabolic pathways, producing antibiotics or other protective compounds. Stationary phase is often marked by a balance between growth and death. Death decline phase. In the death phase, the number of dying cells exceeds the number of new cells being formed. This phase is characterized by nutrient depletion, accumulation of toxic waste products, and a general decline in cellular functions. Microorganisms that cannot adapt to the stressful conditions eventually die. 3. Factors influencing growth yield and characteristics. Several environmental and intrinsic factors influence microbial growth yield and characteristics. Nutrient availability. The type and concentration of nutrients, including carbon, nitrogen, sulfur, and phosphorus, significantly affect microbial growth yield. Nutrient limitation can result in slower growth rates and reduced yield. Oxygen availability. Oxygen availability determines whether a microorganism undergoes aerobic respiration, which yields higher growth yields, or anaerobic respiration fermentation, which often results in lower yields. Temperature. Microorganisms have an optimal temperature range for growth. Too high or too low temperatures can inhibit growth and reduce the yield. pH. The pH of the growth medium affects enzyme activity and membrane integrity. Extreme pH levels can reduce growth rates and decrease microbial yield. Salinity. Salt concentration influences osmotic pressure and can either support or inhibit microbial growth depending on the species' tolerance to salinity. 4. Measurement of growth and yield. Optical density OD. One common method to monitor microbial growth is measuring the optical density at 600 nanometers OD 600 using a spectrophotometer. OD 600 correlates with cell concentration in a sample, providing an estimate of growth. Plate count method. This involves plating diluted samples on agar plates and counting the number of colony forming units CFUs. This method provides a direct count of viable microorganisms. Dry weight measurement. Another method to quantify microbial biomass involves drying the microbial culture and measuring its weight, which reflects the total biomass produced. 5. Carbon and energy yield. The yield can also be related to the efficiency with which microorganisms convert carbon sources, like glucose, into cellular biomass. Carbon source availability directly affects the rate of growth and the final biomass yield. The type of metabolic pathway employed by the microorganism also affects the efficiency of energy production. Fermentation versus respiration. Fermentation typically yields less biomass compared to respiration because it involves the incomplete oxidation of substrates, leading to less ATP production. ATP yield. The yield of ATP during microbial growth depends on the metabolic pathway. Aerobic respiration is more efficient than anaerobic respiration or fermentation in terms of ATP production per molecule of substrate. Previous year questions. Question CSIRNET 2017. Which of the following factors does not significantly influence the yield of microbial growth? A. Nutrient concentration. B. pH of the medium. C. Light intensity. D. Oxygen availability. Answer. C. Light intensity. Question CSIRNET 2019. Which phase of microbial growth is characterized by a constant rate of cell division? A. Lag phase. B. Exponential phase. C. Stationary phase. D. Death phase. Answer. B. Exponential phase. Question CSIRNET 2020. During the stationary phase of microbial growth, the rate of a. Nutrient uptake is maximized. B. Cell division equals the rate of cell death. C. Protein synthesis is at its highest. D. Growth rate exceeds the death rate. Answer. B. Cell division equals the rate of cell death. This lecture on growth yield and characteristics in microbial physiology provides a foundational understanding of how microorganisms grow, how the growth can be measured, and the factors influencing the yield. It is vital for applications in industrial microbiology, biotechnology, and medicine, particularly in optimizing conditions for microbial production of metabolites or bioproducts. You said. Strategies of cell division. ChatGPT said. Strategies of cell division. Cell division is a fundamental process for life, enabling organisms to grow, reproduce, and maintain homeostasis. Different organisms utilize distinct strategies of cell division, depending on the type of organism and the cellular function required. These strategies primarily involve mitosis and meiosis, but there are also variations in specialized forms that cater to specific biological needs, including binary fission in prokaryotes and budding in some eukaryotes. 1. Mitosis. Somatic cell division. Mitosis is the process by which somatic cells, non-reproductive cells, divide to produce two genetically identical daughter cells. It plays a crucial role in growth, development, tissue repair, and asexual reproduction in multicellular organisms. Mitosis ensures that each daughter cell receives an exact copy of the parent cell's genetic material. Phases of mitosis. Prophase. Chromatin condenses into visible chromosomes, and the nuclear envelope begins to break down. The meiotic spindle forms. Metaphase. Chromosomes align at the cell's equator, known as the metaphase plate, with spindle fibers attached to the centromere of each chromosome. Anaphase. Sister chromatids are pulled apart toward opposite poles of the cell by the shortening of spindle fibers. Telophase. Chromatids reach the poles, the nuclear envelope reforms around each set of chromosomes, and the chromosomes begin to decondense. Cytokinesis. The cytoplasm divides, resulting in two distinct daughter cells, each with a full set of chromosomes. Regulation. The cell cycle and mitosis are tightly regulated by checkpoints G1, G2, M checkpoints, and proteins such as cyclins and cyclin-dependent kinases (CDKs). These regulatory mechanisms ensure that cell division occurs accurately and only when the cell is ready. 2. Meiosis. Sexual reproduction. Meiosis is a type of cell division that reduces the chromosome number by half, producing 4 non-identical daughter cells, each with half the number of chromosomes of the parent cell. Meiosis is essential for sexual reproduction, generating gametes, sperm and eggs in animals, spores in plants that, when fertilized, restore the diploid number in the zygote. Phases of meiosis. Meiosis I. Reduction division. Prophase I. Chromosomes condense, and homologous chromosomes pair up, forming tetrads. Crossing over occurs, where homologous chromosomes exchange genetic material, leading to genetic variation. Metaphase I. Homologous chromosome pairs align at the metaphase plate. Anaphase I. Homologous chromosomes are pulled to opposite poles. Unlike mitosis, the sister chromatids remain attached. Telophase I. The nuclear envelope reforms around the two sets of chromosomes. Cytokinesis occurs, resulting in two haploid daughter cells. Meiosis II. Equational division, similar to mitosis. Prophase II. Chromosomes recondense, and the nuclear envelope breaks down. Metaphase II. Chromosomes align at the metaphase plate. Anaphase II. The sister chromatids are separated and move toward opposite poles. Telophase II. The nuclear envelope reforms, and cytokinesis occurs. This results in 4 genetically diverse haploid cells. Genetic variation. Meiosis introduces genetic diversity through processes like crossing over and independent assortment, where homologous chromosomes are shuffled randomly during metaphase I, and chromosomes are sorted into gametes independently of each other. 3. Binary fission. Prokaryotic cell division. Binary fission is the primary mechanism of cell division in prokaryotes, such as bacteria and archaea. Unlike mitosis, binary fission is a relatively simple process that involves the replication of a single, circular DNA molecule and its subsequent division into two daughter cells. Steps in binary fission. DNA replication. The DNA molecule is copied, and each copy attaches to a different part of the cell membrane. Cell elongation. The cell elongates, and the two DNA copies are pulled to opposite ends of the cell. Septum formation. A septum partition begins to form in the middle of the cell. Cytokinesis. The septum fully forms, and the cell is divided into two genetically identical daughter cells. Regulation. In prokaryotes, binary fission is primarily regulated by the availability of nutrients and environmental conditions, as well as the action of proteins like FTSC, which is involved in septum formation. 4. Budding. Asexual reproduction in yeast and other organisms. Budding is a form of asexual reproduction found in certain eukaryotic organisms, such as yeast, hydra, and some plants. In budding, a new organism develops from an outgrowth bud of the parent organism. The bud grows and eventually detaches to become an independent organism. Steps in budding. A small bulge or bud forms on the parent cell or organism. The bud enlarges and undergoes meiotic cell division to produce new cells. Once the bud has matured, it detaches from the parent to become a separate organism. Regulation. Budding in yeast such as Saccharomyces cerevisiae is regulated by nutrient availability and signaling pathways like the ROS-CAM pathway, which controls the progression of the cell cycle in mitosis. 5. Endomitosis polyploidization. Endomitosis is a type of cell division where mitosis occurs without cytokinesis, resulting in the formation of a cell with multiple copies of the genome polyploidy. This is commonly observed in certain specialized cells like megakaryocytes, precursors to platelets, and some cancer cells. Mechanism. The cell undergoes DNA replication and meiotic spindle formation, but the chromosomes are not segregated into daughter cells. Instead, the nuclear membrane reforms around the replicated chromosomes, resulting in a polyploid nucleus. Function and occurrence. Endomitosis is important in producing cells with large amounts of genetic material, such as in megakaryocytes, where it contributes to the production of platelets. 6. Specialized division in the immune system. Clonal expansion. In the immune system, especially in T and B lymphocytes, clonal expansion involves rapid cell division after activation. This process ensures a large number of immune cells are produced in response to an antigen. It's a form of division that occurs following recognition of a pathogen and is crucial for adaptive immunity. Mechanism. Upon encountering a specific antigen, naive T and B cells undergo clonal expansion through meiotic division, producing numerous identical cells that can respond to the antigen. This process also involves differentiation into effector cells and memory cells, ensuring a more robust response upon future infections. 7. Fission in parasitic protozoa. Schizogony. Schizogony is a form of asexual reproduction found in certain protozoa, particularly in the life cycle of some parasitic organisms like Plasmodium, the causative agent of malaria. In schizogony, the cell undergoes multiple rounds of nuclear division before the cytoplasm divides to form several daughter cells. Steps in schizogony. The nucleus divides several times, producing multiple nuclei. The cytoplasm divides, and each nucleus is enclosed in a separate daughter cell. The newly formed cells are released, often infecting new host cells. Previous year questions. Question CSIRNET 2018. In which of the following cell division processes is the chromosome number reduced by half? A. Mitosis. B. Binary fission. C. Meiosis. D. Budding. Answer. C. Meiosis. Question CSIRNET 2017. Which of the following events occurs during prophase I of meiosis? A. Chromosomes align at the metaphase plate. B. Crossing over occurs between homologous chromosomes. C. Sister chromatids are pulled apart. D. Cytokinesis takes place. Answer. B. Crossing over occurs between homologous chromosomes. Question CSIRNET 2020. The process of binary fission in bacteria includes all of the following steps except a. DNA replication. B. Formation of a septum. C. Crossing over of genetic material. D. Division into two identical daughter cells. Answer. C. Crossing over of genetic material. This lecture on the strategies of cell division provides insight into the diversity of mechanisms through which cells reproduce, from simple binary fission in prokaryotes to complex processes like meiosis in eukaryotic organisms. These strategies are essential for the maintenance of life, growth, and reproduction across different kingdoms of life. You said. Microbial physiology strategies of cell division. ChatGPT said. Microbial physiology strategies of cell division. Microbial cell division is a key process in microbial physiology, driving the growth, proliferation, and reproduction of microorganisms. Unlike multicellular organisms that mainly rely on mitosis and meiosis, microbes, particularly prokaryotes, use a variety of division strategies that are adapted to their unicellular nature and environmental conditions. The strategies of cell division in microbes vary significantly based on their type, bacteria, archaea, fungi, and protozoa, and the complexity of their internal structures. The main strategies include binary fission, budding, fragmentation, and sporulation, each of which plays a critical role in microbial growth and survival. 1. Binary fission in bacteria. Binary fission is the most common form of cell division in prokaryotic organisms, particularly in bacteria. It is an asexual process that results in the formation of two genetically identical daughter cells. This process involves the duplication of the bacterial genome, followed by the segregation of the genetic material into two separate daughter cells. Steps in binary fission. DNA replication. The bacterial chromosome, which is typically a single, circular DNA molecule, is replicated. The origin of replication is at the ORIC in E. coli, where the DNA replication machinery begins to copy the genetic material. Elongation. The bacterial cell elongates, and the two daughter chromosomes are separated toward opposite poles of the cell. Septum formation. A protein structure, including FTSC, forms the septum of the midcell, a ring-like structure that divides the cell into two parts. The septum is created through a combination of membrane and peptidoglycan synthesis. Cytokinesis. The septum fully forms, and the cell wall is completed. This results in the division of the cell into two genetically identical daughter cells. Regulation of binary fission. Binary fission is tightly regulated by proteins such as FTSC, which are involved in cell division, and proteins, which ensure that the septum forms at the center of the cell. The rate of division is also controlled by environmental factors like nutrient availability and cell size. 2. Budding in yeast and some bacteria. Budding is an asexual form of reproduction that occurs in certain fungi, such as yeast and some bacteria. In budding, a new cell forms as an outgrowth or bud on the parent cell. This process allows the parent cell to remain intact while producing genetically identical offspring. Steps in budding. Formation of the bud. The parent cell begins to form a small protrusion, or bud, usually at a specific site on the cell membrane. Nuclear division. The nucleus of the parent cell divides, and one copy of the genetic material is directed into the bud. Cell elongation. The bud elongates, and the daughter nucleus is incorporated into it. Separation. The bud eventually separates from the parent cell, resulting in two daughter cells, one of which may remain attached to the parent cell in a chain-like structure, as seen in Saccharomyces cerevisiae. Regulation of budding. In yeast, budding is controlled by the cell cycle regulatory proteins such as cyclins and cyclin-dependent kinases (CDKs). These proteins help coordinate the initiation of DNA replication and mitosis, allowing the bud to develop properly. 3. Fragmentation in cyanobacteria and algae. Fragmentation is a method of cell division seen in some types of algae and cyanobacteria. In this form of asexual reproduction, a parent organism breaks into multiple fragments, each of which grows into a new, independent organism. Steps in fragmentation. The parent organism, e.g., a filamentous cyanobacterium, physically breaks into multiple pieces. Each fragment contains a part of the parent's genetic material, and with the appropriate conditions, nutrients, temperature, each fragment grows into a new, fully functional organism. Regulation. Fragmentation is typically triggered by environmental factors such as nutrient availability or unfavorable conditions that stress the parent organism. The regulation involves a combination of genetic factors and external signals that activate cell division and growth in the fragments. 4. Sporulation in bacteria, e.g., Bacillus and Clostridium. Sporulation is a survival strategy used by certain bacteria, such as Bacillus and Clostridium, to endure unfavorable environmental conditions, such as nutrient depletion or harsh physical conditions. In sporulation, a bacterial cell forms a highly resistant spore that can survive extreme stress and later germinate. Microbial physiology. Strategies of cell division. Microbial cell division is a key process in microbial physiology, driving the growth, proliferation, and reproduction of microorganisms. Unlike multicellular organisms that mainly rely on mitosis and meiosis, microbes, particularly prokaryotes, use a variety of division strategies that are adapted to their unicellular nature and environmental conditions. The strategies of cell division in microbes vary significantly based on their type, bacteria, archaea, fungi, and protozoa, and the complexity of their internal structures. The main strategies include binary fission, budding, fragmentation, and sporulation, each of which plays a critical role in microbial growth and survival. 1. Binary fission in bacteria. Binary fission is the most common form of cell division in prokaryotic organisms, particularly in bacteria. It is an asexual process that results in the formation of two genetically identical daughter cells. This process involves the duplication of the bacterial genome, followed by the segregation of the genetic material into two separate daughter cells. Steps in binary fission. DNA replication. The bacterial chromosome, which is typically a single, circular DNA molecule, is replicated. The origin of replication is at the ORIC in E. coli, where the DNA replication machinery begins to copy the genetic material. Elongation. The bacterial cell elongates, and the two daughter chromosomes are separated toward opposite poles of the cell. Septum formation. A protein structure, including FTSC, forms the septum of the mid cell, a ring-like structure that divides the cell into two parts. The septum is created through a combination of membrane and peptidoglycan synthesis. Cytokinesis. The septum fully forms, and the cell wall is completed. This results in the division of the cell into two genetically identical daughter cells. Regulation of binary fission. Binary fission is tightly regulated by proteins such as FTSC, which are involved in cell division, and min proteins, which ensure that the septum forms at the center of the cell. The rate of division is also controlled by environmental factors like nutrient availability and cell size. 2. Budding in yeast and some bacteria. Budding is an asexual form of reproduction that occurs in certain fungi, such as yeast and some bacteria. In budding, a new cell forms as an outgrowth or bud on the parent cell. This process allows the parent cell to remain intact while producing genetically identical offspring. Steps in budding. Formation of the bud. The parent cell begins to form a small protrusion, or bud, usually at a specific site on the cell membrane. Nuclear division. The nucleus of the parent cell divides, and one copy of the genetic material is directed into the bud. Cell elongation. The bud elongates, and the daughter nucleus is incorporated into it. Separation. The bud eventually separates from the parent cell, resulting in two daughter cells, one of which may remain attached to the parent cell in a chain-like structure, as seen in Saccharomyces cerevisiae. Regulation of budding. In yeast, budding is controlled by the cell cycle regulatory proteins such as cyclins and cyclin-dependent kinases (CDKs). These proteins help coordinate the initiation of DNA replication and mitosis, allowing the bud to develop properly. 3. Fragmentation in cyanobacteria and algae. Fragmentation is a method of cell division seen in some types of algae and cyanobacteria. In this form of asexual reproduction, a parent organism breaks into multiple fragments, each of which grows into a new, independent organism. Steps in fragmentation. The parent organism, e.g., a filamentous cyanobacterium, physically breaks into multiple pieces. Each fragment contains a part of the parent's genetic material, and with the appropriate conditions, nutrients, temperature, each fragment grows into a new, fully functional organism. Regulation. Fragmentation is typically triggered by environmental factors such as nutrient availability or unfavorable conditions that stress the parent organism. The regulation involves a combination of genetic factors and external signals that activate cell division and growth in the fragments. 4. Sporulation in bacteria, e.g., Bacillus and Clostridium. Sporulation is a survival strategy used by certain bacteria, such as Bacillus and Clostridium, to endure unfavorable environmental conditions, such as nutrient depletion or harsh physical conditions. In sporulation, a bacterial cell forms a highly resistant spore that can survive extreme stress and later germinate when conditions become favorable. Steps in sporulation. DNA replication. The bacterium replicates its DNA and begins to form a protective coat around a part of the chromosome. Asymmetric division. The cell divides asymmetrically, creating a larger mother cell and a smaller porospore. Spore formation. The porospore is surrounded by a multi-layered protective coat, and the mother cell deteriorates. Mature spore. The result is a dormant, highly resistant spore that can survive extreme conditions like heat, desiccation, and radiation. Regulation of sporulation. The process is regulated by a complex network of transcription factors such as SPO0A, which acts as a master regulator of sporulation. Environmental factors like nutrient depletion, stress, and high temperature activate these pathways to trigger sporulation. 5. Conjugation. Horizontal gene transfer in bacteria. Though not a form of division, conjugation is a key strategy for gene transfer in bacteria. During conjugation, a donor cell transfers genetic material to a recipient cell through a specialized pilus. Steps in conjugation. Formation of pilus. The donor bacterium extends a pilus to connect with the recipient bacterium. Transfer of genetic material. The donor bacterium transfers a plasmid or a portion of its chromosome to the recipient bacterium through the pilus. Separation. Once the DNA has been transferred, the cells separate, and both now contain the transferred genetic material. Regulation of conjugation. Conjugation is controlled by plasmids and specific genes, such as the TRA genes, which control the formation of the pilus and the transfer of DNA. Environmental conditions, such as the presence of specific signaling molecules, can influence the frequency of conjugation. Key points in microbial cell division. Binary fission is the primary strategy for cell division in prokaryotes, ensuring rapid population growth under favorable conditions. Budding allows for asexual reproduction in yeast and some bacteria, enabling them to proliferate without complete cell division. Fragmentation in some cyanobacteria and algae involves breaking into smaller pieces, which grow into new organisms. Sporulation is a survival strategy in bacteria, leading to the formation of dormant spores that can survive harsh conditions. Conjugation allows bacteria to transfer genetic material, contributing to genetic diversity and evolution. Previous year questions. Question. CSIRNET 2019. Which of the following methods of bacterial cell division involves the formation of a septum at the center of the cell? A. Binary fission. B. Budding. C. Fragmentation. D. Sporulation. Answer. A. Binary fission. Question. CSIRNET 2018. Sporulation in Bacillus and Clostridium results in the formation of A. Asexual spores. B. Vegetative cell. C. Genetic recombination. D. Bacterial endospores. Answer. D. Bacterial endospores. Question. CSIRNET 2020. In which form of microbial reproduction does the parent organism break into several pieces, each capable of developing into a new organism? A. Binary fission. B. Budding. C. Fragmentation. D. Conjugation. Answer. C. Fragmentation. These strategies of microbial cell division reflect the diversity of mechanisms that microbes employ to reproduce, survive, and adapt to their environments. Each method is regulated by specific molecular and environmental signals that ensure the appropriate division process is activated based on the needs of the organism. You said. Microbial physiology stress response. ChatGPT said. Microbial physiology stress response. Microorganisms are continuously exposed to various environmental stresses, such as temperature fluctuations, oxidative stress, nutrient limitation, osmotic pressure, and exposure to toxic compounds. In order to survive and thrive under such harsh conditions, microbes have evolved complex stress response mechanisms that enable them to sense and adapt to changes in their environment. These responses are vital for microbial survival and are particularly important in pathogenesis, biofilm formation, and antibiotic resistance. The stress response mechanisms can be broadly categorized into general stress responses and specific stress responses, each of which is tightly regulated by specific proteins and signaling pathways. 1. General stress response. The general stress response refers to the set of adaptive strategies that microorganisms employ when they encounter a wide range of environmental stressors. The main regulatory mechanisms are sigma-S, sigma-S or RPOs in bacteria such as Escherichia coli. The general stress response is largely controlled by the alternative sigma factor sigma-S RPOs, which is part of the RNA polymerase complex. Under stress conditions such as nutrient starvation or oxidative stress, the RPO sigma factor is upregulated, leading to the activation of stress-responsive genes. Role. Sigma-S regulates the expression of genes involved in protective functions like detoxification, membrane maintenance, and repair of cellular structures. It also helps in the modulation of metabolic pathways to adapt to the stressor. Mechanism. Under normal conditions, RPOs is degraded by the CLPXP protease. During stress, phosphorylation of RPO stabilizes it, allowing it to associate with RNA polymerase and initiate transcription of stress-related genes. Heat shock response. The heat shock response is one of the most conserved stress responses in microbes and is activated by elevated temperatures or other stresses that can denature proteins. Heat shock proteins HSPs. The heat shock response involves the induction of a class of proteins known as heat shock proteins HSPs. These proteins function as molecular chaperones, helping in the refolding of denatured proteins and preventing aggregation. Key proteins. In bacteria, the major heat shock protein is DNAC HSP70, which binds to misfolded proteins and helps in their refolding. Other proteins like ROL, chaperonins, and CLPP are also involved in this process. Regulation. The heat shock response is primarily controlled by the sigma-32 RPOH sigma factor, which induces the expression of heat shock genes when cells are exposed to elevated temperatures. 2. Oxidative stress response. Oxidative stress is caused by an imbalance between reactive oxygen species, ROS, and the cell's ability to detoxify them. ROS can damage cellular components like DNA, proteins, and lipids. To cope with oxidative stress, microorganisms produce antioxidant enzymes and other protective molecules. ROS detoxification enzymes. Superoxide dismutase SOD converts superoxide radicals O into hydrogen peroxide HO. Catalase and peroxidase. These enzymes decompose hydrogen peroxide into water and oxygen, reducing the oxidative damage caused by ROS. Theoretics and glutathione systems. These systems help in the reduction of oxidative damage to proteins and maintain cellular redox balance. Regulation of oxidative stress response. The regulation of oxidative stress responses is mediated by transcription factors such as OXIR in E. coli and SOXARs in E. coli and other gram-negative bacteria. These transcription factors sense the presence of ROS and activate the expression of genes encoding antioxidant enzymes. OXIR. In response to HO, OXIR undergoes oxidation, leading to a conformational change that allows it to bind to DNA and activate the transcription of antioxidant genes. 3. Osmotic stress response. Microorganisms often face osmotic stress due to changes in the surrounding osmolarity, such as high salt concentrations or low water activity. In response, cells must adjust their internal osmotic pressure to maintain cell integrity. Compatible solutes. To balance osmotic pressure, microbes accumulate small, highly soluble organic compounds called compatible solutes, e.g., glycine, betaine, trehalose, and proline. These solutes do not interfere with cellular metabolism and help in stabilizing proteins and maintaining cellular functions under osmotic stress. Osmoregulation pathways. In E. coli, the NF OMPR system regulates the expression of outer membrane proteins in response to osmotic changes. NF senses osmotic stress and activates OMPR, which regulates the transcription of the OMPC and OMPF genes involved in outer membrane porin synthesis. Regulation. Osmotic stress is regulated by several signal transduction pathways, including the two-component system, e.g., NF OMPR, and by osmotic sensors such as KDPD, KDPD, which regulate potassium uptake. 4. Nutrient limitation and starvation response. Microbes often face nutrient limitation, particularly for carbon, nitrogen, or phosphorus. In response, they initiate starvation responses to survive prolonged periods without sufficient nutrients. Starvation survival pathways. Phoregulin. In response to phosphate starvation, bacteria like E. coli activate the phoregulin, which is controlled by the PHO4 two-component system. This system enhances the uptake of phosphate and the expression of phosphate. Microorganisms are continuously exposed to various environmental stresses, such as temperature fluctuations, oxidative stress, nutrient limitation, osmotic pressure, and exposure to toxic compounds. In order to survive and thrive under such harsh conditions, microbes have evolved complex stress response mechanisms that enable them to sense and adapt to changes in their environment. These responses are vital for microbial survival and are particularly important in pathogenesis, biofilm formation, and antibiotic resistance. The stress response mechanisms can be broadly categorized into general stress responses and specific stress responses, each of which is tightly regulated by specific proteins and signaling pathways. 1. General stress response. The general stress response refers to the set of adaptive strategies that microorganisms employ when they encounter a wide range of environmental stressors. The main regulatory mechanisms are sigma-S, sigma-S or RPOs. In bacteria such as Escherichia coli, the general stress response is largely controlled by the alternative sigma factor sigma-S RPOs, which is part of the RNA polymerase complex. Under stress conditions such as nutrient starvation or oxidative stress, the RPO sigma factor is upregulated, leading to the activation of stress-responsive genes. Role. Sigma-S regulates the expression of genes involved in protective functions like detoxification, membrane maintenance, and repair of cellular structures. It also helps in the modulation of metabolic pathways to adapt to the stressor. Mechanism. Under normal conditions, RPOs is degraded by the CLPXP protease. During stress, phosphorylation of RPOs stabilizes it, allowing it to associate with RNA polymerase and initiate transcription of stress-related genes. Heat shock response. The heat shock response is one of the most conserved stress responses in microbes and is activated by elevated temperatures or other stresses that can denature proteins. Heat shock proteins HSPs. The heat shock response involves the induction of a class of proteins known as heat shock proteins HSPs. These proteins function as molecular chaperones, helping in the refolding of denatured proteins and preventing aggregation. Key proteins. In bacteria, the major heat shock protein is DNAC HSP70, which binds to misfolded proteins and helps in their refolding. Other proteins like ROL, chaperonins, and CLPP are also involved in this process. Regulation. The heat shock response is primarily controlled by the sigma-32 RPOH sigma factor, which induces the expression of heat shock genes when cells are exposed to elevated temperatures. 2. Oxidative stress response. Oxidative stress is caused by an imbalance between reactive oxygen species, ROS, and the cell's ability to detoxify them. ROS can damage cellular components like DNA, proteins, and lipids. To cope with oxidative stress, microorganisms produce antioxidant enzymes and other protective molecules. ROS detoxification enzymes. Superoxide dismutase SOD converts superoxide radicals O into hydrogen peroxide HO. Catalase and peroxidase. These enzymes decompose hydrogen peroxide into water and oxygen, reducing the oxidative damage caused by ROS. Theoretics and glutathione systems. These systems help in the reduction of oxidative damage to proteins and maintain cellular redox balance. Regulation of oxidative stress response. The regulation of oxidative stress responses is mediated by transcription factors such as OXIR in E. coli and SOXARs in E. coli and other gram-negative bacteria. These transcription factors sense the presence of ROS and activate the expression of genes encoding antioxidant enzymes. OXIR. In response to HO, OXIR undergoes oxidation, leading to a conformational change that allows it to bind to DNA and activate the transcription of antioxidant genes. 3. Osmotic stress response. Microorganisms often face osmotic stress due to changes in the surrounding osmolarity, such as high salt concentrations or low water activity. In response, cells must adjust their internal osmotic pressure to maintain cell integrity. Compatible solutes. To balance osmotic pressure, microbes accumulate small, highly soluble organic compounds called compatible solutes, e.g., glycine, betaine, trehalose, and proline. These solutes do not interfere with cellular metabolism and help in stabilizing proteins and maintaining cellular functions under osmotic stress. Osmoregulation pathways. In E. coli, the NF OMPR system regulates the expression of outer membrane proteins in response to osmotic changes. NF senses osmotic stress and activates OMPR, which regulates the transcription of the OMPC and OMPF genes involved in outer membrane porin synthesis. Regulation. Osmotic stress is regulated by several signal transduction pathways, including the two-component system, e.g., NF OMPR, and by osmotic sensors such as KDPD, which regulate potassium uptake. 4. Nutrient limitation and starvation response. Microbes often face nutrient limitation, particularly for carbon, nitrogen, or phosphorus. In response, they initiate starvation responses to survive prolonged periods without sufficient nutrients. Starvation survival pathways. Phoregulin. In response to phosphate starvation, bacteria like E. coli activate the phoregulin, which is controlled by the PHO4 two-component system. This system enhances the uptake of phosphate and the expression of phosphate utilization genes. Carbon starvation. In response to carbon limitation, bacteria activate the CSR system, carbon storage regulator, to promote the use of stored carbon sources, and genes involved in catabolic pathways are upregulated. Mechanisms of adaptation. Formation of endospores. In extreme nutrient limitation, some bacteria form highly resistant endospores, e.g., Bacillus species, to survive until conditions improve. Autophagy. In more complex organisms like yeast and fungi, autophagy is activated during nutrient deprivation, where the cell degrades its own components to recycle nutrients. 5. Acid stress response. Microorganisms in acidic environments, e.g., stomachs or acid mine drainage, activate acid stress response mechanisms to protect themselves from low pH. Proteins involved in acid resistance. F0, F1 ACASs. These enzymes pump protons out of the cell to maintain intracellular pH. Acid shock proteins. These proteins help stabilize cellular structures under acidic conditions. Regulation. In E. coli, the HNS protein and the RPO sigma factor help regulate genes involved in acid resistance. Key points on microbial stress responses. General stress response, governed by sigma S RPOs in bacteria, which activates genes for survival under various stresses. Oxidative stress, managed by antioxidant enzymes like SOD, catalase, and regulatory proteins like OXIR. Osmotic stress, microbes use compatible solutes to adapt, and regulatory systems like ENIPs, OMPR control osmotic stress responses. Nutrient limitation, pathways like the thyroglin and autophagy are triggered by nutrient starvation. Acid stress, acid resistance is managed by ACASs and acid shock proteins, with regulators like HNS and E. coli. Previous year questions. Question, CSIRNET 2020. Which of the following stress responses in bacteria involves the induction of heat shock proteins such as DNAC? A heat shock response. B. Oxidative stress response. C. Nutrient stress response. D. Acid stress response. Answer, a heat shock response. Question, CSIRNET 2019. Which of the following stress responses in bacteria involves the induction of heat shock proteins such as DNAC? A heat shock response. B. Oxidative stress response. C. Nutrient stress response. D. Acid stress response. Answer, a heat shock response. Question, CSIRNET 2019. The alternative sigma factor sigma S RPOs in bacteria is primarily involved in a response to heat shock. B. Response to oxidative stress. C. General stress response. D. Response to acid stress. Answer, C. General stress response. Question, CSIRNET 2018. Which of the following systems is involved in bacterial acid resistance by pumping protons out of the cell? A. F0, F1 ACPase. B. Catalase. C. Superoxide dismutase. D. OXIR. Answer, A. F0, F1 ACPase. Understanding these stress responses is crucial for comprehending how microorganisms survive adverse conditions and adapt to changes in their environment. These mechanisms are essential for their survival in diverse habitats, including extreme environments, and are often exploited in biotechnology and medicine. You said. Fundamental processes. A. DNA replication. ChatGPT said. Fundamental processes. DNA replication. DNA replication is a process by which a cell duplicates its DNA to ensure that each daughter cell receives an identical copy of the genetic material. This process is critical for cell division and is highly regulated to maintain the integrity of the genetic information. DNA replication occurs during the S phase of the cell cycle, and it involves a series of highly coordinated steps involving multiple enzymes and proteins to unwind, copy, and reassemble the DNA molecule. 1. Initiation of DNA replication. DNA replication begins at specific locations on the DNA molecule known as origins of replication. In eukaryotes, these origins are numerous and are scattered across the chromosomes, while prokaryotes, such as Escherichia coli, typically have a single origin of replication. Origin recognition. The process starts with the binding of initiator proteins to the origin of replication. In E. coli, the initiator protein is DNA A, which binds to the DNA A boxes in the origin of replication or causing the DNA to unwind locally. Helicase loading. The DNA C protein helps load the DNA B helicase onto the unwound DNA region. Helicase is responsible for unwinding the double-stranded DNA into single-stranded regions, forming a replication bubble. Single-strand binding proteins SSB. Once the DNA is unwound by helicase, single-strand binding proteins SSBs bind to the single-stranded DNA to stabilize it and prevent reannealing of the strands. Primase activity. The enzyme primase RNA polymerase synthesizes a short RNA primer on each of the single strands, providing a starting point for DNA synthesis. In E. coli, this is done by the DNA G primase. 2. Elongation of DNA replication. Once the primers are laid down, DNA polymerase can begin adding nucleotides to the grown DNA strand. This is where the actual synthesis of the new DNA strands occurs. DNA polymerase III. The main enzyme responsible for DNA synthesis is DNA polymerase III in prokaryotes. In eukaryotes, this is carried out by DNA polymerase delta and DNA polymerase epsilon. DNA polymerase III adds nucleotides complementary to the template strand in the 5' to 3' direction. It uses the RNA primer as a starting point to synthesize a new DNA strand. Leading strand. On the leading strand, DNA polymerase III synthesizes a new strand continuously in the direction of the replication fork. Lagging strand. On the lagging strand, DNA replication occurs discontinuously in the form of Okazaki fragments, which are small segments of DNA synthesized in the 5' to 3' direction but away from the replication fork. Each Okazaki fragment requires a new RNA primer to begin synthesis. Sliding clamp. The sliding clamp, also known as the clamp loader complex in eukaryotes, is a protein that ensures DNA polymerase remains attached to the DNA template during replication. In E. coli, the sliding clamp is a clamp loader composed of the tau and gamma subunits, and the clamp the beta clamp, which encircles the DNA and increases the processivity of DNA polymerase. 3. Termination of DNA replication. In prokaryotes like E. coli, replication termination occurs when the two replication forks meet at the terminus of replication tear sites. The termination process involves the 2' protein, which binds to the tear sites and prevents further unwinding of the DNA, effectively halting DNA replication. In eukaryotic cells, termination is more complex and involves the telomerase enzyme, especially in the replication of the ends of chromosomes telomeres. The telomerase extends the telomeres, preventing the loss of genetic material during replication. DNA ligase. After the Okazaki fragments are synthesized, they must be joined together. This is accomplished by the enzyme DNA ligase, which forms phosphodiester bonds between the fragments, resulting in a continuous strand. Proofreading and error correction. DNA polymerases have an inherent proofreading ability due to their 3' to 5' exonuclease activity, which allows them to remove incorrectly incorporated nucleotides and replace them with the correct ones. This is crucial for maintaining high fidelity during DNA replication. 4. Regulation of DNA replication. The process of DNA replication is highly regulated to ensure that the genome is accurately and completely duplicated. Various checkpoint mechanisms exist to prevent replication errors or premature replication before the cell is ready to divide. Cyclins and CDKs. In eukaryotic cells, the initiation of DNA replication is regulated by cyclins and cyclin-dependent kinase CDKs. Cyclins activate CDKs, which in turn phosphorylate key proteins involved in replication initiation. This ensures that replication only occurs once per cell cycle. Origin licensing. Before DNA replication begins, the origin of replication must be licensed. This involves the binding of the origin recognition complex (ORC), which marks the origin as ready for replication. Once replication begins, the origin is fired, and the licensing factors are removed to prevent re-replication. 5. Key enzymes and proteins involved in DNA replication. Helicase. DNA B unwinds the DNA double helix at the replication fork. Primase DNA G synthesizes RNA primers to initiate DNA replication. DNA polymerase III, main enzyme that synthesizes the leading and lagging strands. Sliding clamp beta clamp increases the processivity of DNA polymerase. DNA ligase seals the gaps between Okazaki fragments. Topoisomerase's DNA gyrase relieves torsional strain ahead of the replication fork. Single strand binding proteins SSBs stabilize single stranded regions of DNA. 6. Mechanisms to ensure replication fidelity. Proofreading by DNA polymerase. DNA polymerase has an inherent ability to check its work by removing incorrectly paired bases. Mismatch repair. Post-replication mismatch repair mechanisms correct errors that escape the proofreading function of DNA polymerase. Previous year questions. Question, CSIRNET 2022. Which of the following enzymes is responsible for relieving the torsional strain generated during DNA replication? A. DNA polymerase. B. Topoisomerase. C. Helicase. D. Ligase. Answer, B. Topoisomerase. Question CSIRNET 2021. In DNA replication, the enzyme responsible for synthesizing RNA primers is a DNA polymerase I. B primase C ligase D helicase. Answer, B primase. Question CSIRNET 2020. Which protein binds to the origin of replication and initiates DNA replication in E. coli? A DNA A. B DNA B C primase. D DNA polymerase 3. Answer, a DNA A. Conclusion. DNA replication is an essential process that ensures genetic information is accurately passed on to the daughter cells during cell division. It involves a complex network of proteins and enzymes that work together to unwind, synthesize, and proofread the DNA strands. The regulation of DNA replication is tightly controlled to ensure genomic stability, and the fidelity of this process is crucial for maintaining the integrity of the organism's genetic material. You said. Unit of replication ChatGPT said. Fundamental processes. Unit of replication replicon CSIRNet JRF Life Sciences level. The unit of replication, also known as a replicon, is a segment of DNA that is replicated from a single origin of replication and is controlled by the regulatory sequences associated with that origin. This concept is central to understanding how DNA replication is initiated, progresses, and terminates across both prokaryotic and eukaryotic genomes. 1. Definition and concept of replicon. A replicon is defined as a DNA molecule or region of DNA that contains an origin of replication and all the sequences necessary for the initiation and regulation of replication. It is a functional unit within the genome where DNA replication begins and proceeds bidirectionally or unidirectionally in some cases until the entire replicon is copied. In simple terms, each replicon functions independently, and multiple replicons may be activated simultaneously, especially in large genomes. 2. Replicon in prokaryotes. In most prokaryotes, such as Escherichia coli, the entire circular chromosome is typically a single replicon. Single origin. The chromosome has one origin of replication known as auric. Unidirectional or bidirectional replication. Though replication starts at one origin, it proceeds in both directions. Bidirectional replication, forming two replication forks. The process involves initiator proteins such as DNA A, which binds to the DNA A box sequences at auric and initiates strand separation. Single replicon system ensures tightly coordinated and regulated replication, crucial for the rapid cell division of prokaryotes. 3. Replicon in eukaryotes. In eukaryotic cells, due to the large and linear nature of their chromosomes, each chromosome contains multiple replicons, sometimes thousands per chromosome. Multiple origins. Eukaryotic DNA replication requires multiple origins of replication to ensure the entire genome is copied efficiently within the limited duration of the S phase. Replication timing program. Not all replicons fire at once. Eukaryotes follow a replication timing program, where some replicons are early firing and others are late firing, depending on their location, e.g., euchromatin versus heterochromatin. Autonomously replicating sequences. Rs in Saccharomyces cerevisiae, yeast. The replicon contains Rs, which acts similarly to auric in prokaryotes. Each replicon in eukaryotes is under the control of a licensing mechanism involving proteins like origin recognition complex (ORC), CDC6, CDT1, MCM helicase. These proteins together form the pre-replication complex (pre-RC), which licenses the replicon for one round of replication per cell cycle. 4. Replicon model Jacob, Brenner, and Cousin model. The replicon model was first proposed by Jacob, Brenner, and Cousin 1963 based on studies in E. coli. It suggested the existence of a cis-acting origin, ORI, that acts as a site of replication initiation. A trans-acting initiator protein that specifically recognizes the origin. This model laid the foundation for understanding replication regulation in both prokaryotes and eukaryotes. 5. Control and regulation of replicon. In prokaryotes, replication of a single replicon is tightly regulated through mechanisms like DNA regulation, methylation, e.g., DAM methylase, and feedback control to prevent reinitiation. In eukaryotes, regulation ensures each replicon is activated only once per cell cycle using CDK cyclin-dependent kinases, geminin, which inhibits relicensing, proper degradation and synthesis of licensing factors. This prevents rereplication, a potentially mutagenic process. 6. Features of a replicon. Origin of replication, ORI, the specific site where replication begins. Initiator proteins, recognize the ORI and recruit other factors. Regulatory elements, control the timing, frequency, and fidelity of replication. Bidirectional forks, replication usually proceeds in both directions from the origin. Termination region, site where replication forks converge and DNA synthesis ends. 7. Replicon size and number. In E. coli, the replicon is approximately 4.6 megabits in size entire genome. In eukaryotes like humans, replicons are approximately 50 to 300 kb, and human cells contain 30,000 - 50,000 replicons per S phase. 8. Special cases. Mitochondrial DNA replicons replicate independently with their own origins and machinery. Plasmids, extrachromosomal replicons with their own ORI, e.g., col1 origin and cloning vectors. Viral replicons. Viral genomes act as replicons once inside the host cell, utilizing host or viral enzymes. Previous year questions. CSIRNet question. CSIRNet Deck 2020. In the replicon model proposed by Jacob et al., which one of the following functions is a trans-acting factor? A. Origin of replication. B. Operator. C. Repressor. D. Initiator protein. Answer. D. Initiator protein. Question CSIRNet June 2019. Which of the following best describes a replicon? A region of DNA that codes for a protein. B. A single-stranded RNA molecule. C. A segment of DNA with its own origin of replication. D. A region of DNA that terminates transcription. Answer. C. A segment of DNA with its own origin of replication. Question CSIRNet Deck 2022. In eukaryotic replication, which of the following protein complexes is responsible for loading MCM helicase onto the origin? A. ORC. B. CDT1 CDC6 C. CDKD RPA. Answer. B. CDT1 CDC6. Final notes. The replicon concept is central to understanding how cells replicate their DNA faithfully and only once per cycle. Prokaryotic genomes typically function as a single replicon with simple control, whereas eukaryotic genomes are divided into thousands of replicons, each requiring complex and tightly controlled regulation. Understanding the unit of replication is crucial for advanced studies in molecular biology, genetic engineering, and genome stability, all of which are frequent question areas in CSIRNet Life Sciences exams. You said. Enzymes involved. ChatGPT said. Fundamental processes. Enzymes involved in DNA replication. CSIRNet JRF Life Sciences level, from base to advanced. DNA replication is a fundamental biological process essential for the inheritance of genetic information during cell division. This process is tightly regulated and highly coordinated, involving a suite of enzymes that ensure accurate and efficient synthesis of a new DNA strand. The core objective of DNA replication is to produce two identical copies of a DNA molecule from the parent DNA strand, ensuring genetic fidelity. The entire process follows a semi-conservative model, where each daughter DNA receives one parental and one newly synthesized strand. 1. Helicase, DNA unwinding. Function. Helicases are responsible for unwinding the double-stranded DNA into single strands by breaking the hydrogen bonds between base pairs of the replication fork. In prokaryotes, the helicase is DNAB. In eukaryotes, the helicase complex is part of the MCM, mini-chromosome maintenance complex, composed of MCM2 to 7 proteins, and is loaded at the origin by CDC6 and CDT1 with help from ORC. Mechanism. These enzymes utilize ATP hydrolysis to catalyze the unwinding of DNA. Helicase activity generates supercoiling ahead of the fork, which is resolved by topoisomerases. 2. Single-stranded binding proteins, SSB, RPA. Function. Once the DNA is unwound, single-stranded DNA binding proteins, SSBs, stabilize the exposed strands to prevent reannealing and protect them from nucleases. SSB in prokaryotes. RPA, replication protein A in eukaryotes. These proteins play a crucial role in maintaining the single-stranded structure necessary for DNA polymerase function. 3. Topoisomerase. Relieving supercoiling. Function. As helicase unwinds DNA, it introduces positive supercoil.